relatively short video here to kind of talk about how bacteria eat, right? Bacteria are prokaryotes, and so they basically need to get stuff from the outside environment across their cell membrane, their lipid bilayer, and into their interior, which is just a giant soup of biochemical sort of processes and enzymes and reactions to be able to do a lot of the same biochemistry that we do in terms of fuel metabolism. So one of the ways that they're able to do that is through substances called porins. So porins are essentially a bacterial transport protein. So a bacterial transport protein. And in this case, we are in the class of thinking about passive mediated transport. So we need assistance from these porins to move something with its concentration gradient from high concentration to low. So specifically what we're talking about is related to this structure that we see here. Hopefully you remember this is the structure of starch. Remember what starch is. Starch is glucose, is a homopolymer of glucose, specifically if we're talking about alpha amylose, that's unbranched starch. So it is a homopolymer of glucose that is linked with alpha 1,4 glycosidic bonds. So just a good refresher and reminder of this. So this is alpha amylose. So alpha amylose, it is unbranched homopolymer of glucose with alpha 1,4 glycosidic bonds. Okay, and I'm glad I still have my molecule here of glucose to sort of talk about. So importantly, when we have that alpha 1,4 glycosidic bond, that creates a repeating sort of backbone geometry, if you will, that creates the coil that we see with starch. Remember, this is why we talked about when you put iodine in starch, it will turn blue-black, because you sort of change the electronic properties that we have for iodine, which is able to sort of bind inside the center of this cyclic sort of coiled structure. When we have beta 1,4 glycosidic linkages, that's what we find in cellulose, that tends to form sheets, which is important not only for the function of cellulose, kind of having a more structural protective role, but it also allows it to not interfere with that starch piece. You're going to have to come get her because she's not going to come on her own. Charlotte might come on her own. Well, hello, puppers.
Video?
I'm recording, yes.
Sorry. All right, come on.
So one key thing that we're going to talk about on this video that's going to have a lot of connections to other things that we'll talk about is when you have large molecules that you need to get across a lipid bilayer, how do you do that and have a gate, if you will, that's big enough to hold your big molecule but not let lots of small things get through, okay? So one way that we'll talk about in another video with the potassium channel is how do you get potassium through the bigger basketball and not let sodium through the smaller baseball, right? So until crystal structures were solved, didn't really fully understand that, Nobel Prize awarded for that. What we're going to talk about here is how you have a piece of starch that's corkscrew shaped. How do you actually have a hole that's big enough to allow that, but not let everything else literally pour in? And so we're going to be able to understand that when we look at the structures that are within, or the amino acids that are within sort of the hole that we have here. So I want to highlight, and let me highlight this with some color here. Again, here is the backbone that we have for starch, right? We're going to have this corkscrew shape that we have here. All of those carbon atoms that we can see, we're going to show in green here. So we've got all of those carbon atoms that we're going to have in green here. And importantly, we're going to have all of the red oxygens that are on the periphery. Let's come back over here. Okay, do you see how there's all of these red oxygens that are at the periphery? I'm glad I have my model here still. Okay, this is my model of glucose, right? We've used this for other things. So here's glucose. Here's the oxygen that's in the ring. When we have beta-D-glucose, remember we've got all of our important substituents in equatorial positions, okay? But what I want to do now is I want to have you look at glucose like this, right? And what we can see here, all in the center here is going to be our carbon atoms, And then all on the periphery are going to be our oxygen atoms, right? All of these oxygen atoms on the periphery. So I want you to think of this as a ribbon that is nonpolar where the carbons are, and then polar on the sides. Almost like if you had a ribbon that had like little beads or something on the sides of the ribbon, those beads would be the hydrophilic oxygen atoms. And then the center of the ribbon would be my greasy carbon atoms. So that's how I want you to think about that. We can sort of see that when we look at the structure here. Okay, we've got the greasy black part that forms the bulk of the ribbon, and then we've got all of these red oxygen atoms that are on the side of the ribbon. We can see that when we zoom in and we look at this structure here. Okay, we can see that we've got all of these kind of red oxygen atoms that are on the periphery. And look at what we have in the enzyme active site. So I'm going to highlight here. What we have here in green, okay, is our corkscrew starch, right? Which, to highlight, we're going to have a greasy strip in the center. Okay, then I'm going to change colors here because when we talk about those red oxygen atoms, we're going to have a hydrophilic side to that ribbon. Those are going to be our oxygen atoms. Okay, so if we look inside this enzyme active site, what we see are a whole bunch of, actually got to zoom out here so we get all of them, a whole bunch of aromatic greasy residues. Okay, so this is all a very large aromatic set of amino acids, and this is going to be, again, what we're going to refer to as a greasy slide. A greasy slide. Okay, so you've got a greasy ribbon and a greasy slide. They are going to be happy when they find one another. Okay, this is going to introduce some pieces that we'll sort of talk about in our potassium sort of channel as well. When we have a piece of starch that's in water, okay, aqueous environment, water is very nonpolar. What we're going to see is we are going to see a lot of water that's interacting with those oxygen atoms, okay? So what we're going to need to do to get this corkscrew piece of starch through our porin channel is we need to remove those water molecules. I need you to remember that that is an enthalpic penalty. It's enthalpically unfavorable to break those interactions. As long as we get something back and can form a new interaction, we're going to be okay. So what happens is we are going to dehydrate water from our little piece of starch here, and then we're going to be able to thread it through this channel. Now, when the greasy strip on our corkscrew starch interacts with the greasy slide of our sort of channel through here, favorable London dispersion forces, those nonpolar interactions, are going to have an enthalpic compensation. We're going to get our penalty paid for by making new interactions. Now, you might sort of say, well, why is there like not stuff just pouring through this channel all the time? Isn't that why they're called porins? No, it's not. It is a greasy interior there. Water is totally repelled. It wants nothing to do with that. So even though, and it's not just like it's a gaping hole, but it is a slit, if you will, that's very greasy on the inside. So anything that's aqueous is not going to want to hang out in there. But again, if we strip the water off of this corkscrew, what we can do is we can have that greasy strip interact with our greasy slide, and we can corkscrew through our piece of starch in so that our bacteria can eat. All right, that's how porins work to bring in starch to bacterial cells.